What if you could edit the very code of life? I'm not talking about science fiction, I'm talking about right here, right now. Today, we're diving deep into CRISPR-Cas9. It's a revolutionary technology that, believe it or not, started as a humble immune system in bacteria and has since become one of the most powerful and, yeah, one of the most controversial tools in all of science. So let's get into how this incredible thing works, what it promises, and the massive questions it's forcing all of us to ask.
All right, so here's our roadmap. We're going to start with its super surprising origins out in nature. Then, we'll see how scientists basically hacked it to create this amazing gene editing tool. And we'll look at its incredible promise for medicine. After that, though, we have to get real about the serious technical dangers and the ethical firestorm that erupted with the CRISPR babies. And finally, we'll tackle the biggest question of all. Where do we possibly go from here? You'd think this story starts in some shiny, futuristic lab, right? Well, no, our story actually begins with some of the simplest life forms on Earth, bacteria. Because at its core, CRISPR is an ancient and frankly an incredibly clever defense system that bacteria evolved to fight off viruses. So think about this for a second. How does a simple bacterium, which has been around for billions of years, manage to develop an immune system that has a memory? How can it possibly recognize a virus that's attacked it before and then fight it off the next time? Well, the answer to that is the CRISPR system. Probably the easiest way to wrap your head around this is to picture a vaccination card, or maybe like a most wanted gallery for viruses. When a virus attacks, the bacterium doesn't just try to survive. It actually takes notes so it's ready for the next encounter. So this whole process breaks down into three really brilliant stages. First up is adaptation. The bacterium literally grabs a little piece of the invading virus's DNA and stitches it right into its own genetic code. Step 2 is processing. It makes RNA copies from these stored viral mugshots, and then step 3, interference. These little RNA guides act like a GPS, leading a protein assassin, we'll talk more about that in a sec, directly to any new invaders. It finds the matching sequence and, snip, it just cuts the viral DNA to ribbons. It's a fantastic, self-updating defense system. And this, right here, is where the story takes a huge turn. This is where human ingenuity comes in. Scientists looked at this amazing natural defense and had a revolutionary thought. What if we could reprogram it? What if we could give it our own mugshot and tell it to find and cut any DNA sequence we wanted? And when I say this happened fast, I mean lightning fast. In 2012, researchers Emmanuel Charpentier and Jennifer Doudna figured out how to streamline the whole system. Just 1 year later, 1 year, it was already being used to edit genes in human cells. And by 2020, their work was awarded the Nobel Prize. We're talking less than a decade from basic research to one of the highest honors in science. That's just wild. So what was the secret sauce? What was the big hack that made this so powerful? It was all about making it simpler. See, in nature, the system needed 2 different RNA molecules to work. The big breakthrough was figuring out how to fuse them into just one single guide RNA. And that one change made the tool incredibly easy to program. You just change the code in that one little piece of RNA, and you can send the cutting machine anywhere you want. And that brings us to the real star of the show, the Cas9 protein. This is the enzyme that does the actual cutting. Honestly, the best way to think of it is as a pair of molecular scissors. The guide RNA is the GPS that tells the scissors exactly where in our 3 billion letter long genome to go and make a perfect, precise cut. So once you have a programmable tool that can cut DNA with that kind of precision, well, the possibilities just seem endless. This wasn't just some new toy for researchers. It felt like the beginning of a whole new era for medicine. It offered the potential to treat, maybe even cure genetic diseases that we once thought were completely untouchable. I mean, just look at the sheer breadth of its potential here. We're talking about going after diseases at their absolute source, the genetic code itself. In labs, scientists have used CRISPR to correct the gene for sickle cell anemia, to literally cut out the mutation that causes muscular dystrophy, and even to attack viruses like HIV right where they hide inside our own DNA. And the potential goes way beyond medicine too, with amazing research into engineering crops that can fight off diseases all on their own. And this isn't just hype. This quote really gets to the heart of it. It's not some distant dream. The fact that CRISPR is so simple, so precise, and so efficient is what's making the idea of treating genetic diseases at their source a totally feasible, totally tangible goal. Okay, so it all sounds amazing, right? A medical revolution. But, and this is a huge but, with great power comes great responsibility, and of course, great risk.
Editing the human genome, especially in a way that can be passed down for generations, is not something you do lightly. And this tool, as powerful as it is not perfect.
So let's walk through the 4 big nightmares that keep scientists up at night. First, you've got off-target mutations. Basically, the molecular scissors cut in the wrong place in the genome, and who knows what that could do. Second is mosaicism, where the edit doesn't work in every single cell, creating a person who's a literal patchwork of edited and unedited genes. Third, large deletions, where the tool doesn't just make a clean snip, but accidentally gouges out a huge chunk of DNA, maybe taking vital genes with it. And finally, on-site damage, which is kind of like friendly fire, where you actually damage the healthy gene while trying to fix the broken one.
And to put that risk into perspective, this isn't just a theoretical worry. A major study in rodents found that nearly 30% of the gene-edited animal lines they created had these off-target mutations. We're talking unintended random changes scattered all over their DNA with completely unpredictable results. Mosaicism is also a massive problem. Imagine you're trying to fix a typo in a book, but you only manage to fix it on some of the pages. The result is a total mess. The same thing can happen in an embryo. This means the therapy might not work, or it could have bizarre, unforeseen effects because the person is this genetic mix. It just makes the outcome dangerously unpredictable. So all these risks were very well known in the scientific community. The consensus was pretty clear. We need way more research before this tech is ever used on human embryos that are actually going to become people. But in 2018, that line was crossed. In November of 2018, the world was absolutely stunned by an announcement from a scientist named He Jiankui. He claimed that he had used CRISPR to edit the genomes of twin baby girls Lulu and Nana when they were just single-cell embryos. His stated goal was to disable a gene called CCR5, which would theoretically make the girls resistant to HIV infection. And when you look at what actually happened, it was a complete mess. For Lulu, the edit created 2 brand new mutations that nobody had ever seen before, with totally unknown effects. Plus, they found an off-target cut. For Nana, one copy of the gene was mutated in a new way, but the other copy was left completely untouched. So the main goal of providing HIV immunity, it failed. And both twins were left with permanent, novel mutations in their DNA. This wasn't some clean success story. It was a real-world demonstration of all those risks we just talked about. The reaction from the global scientific community was not mixed. It was swift and it was furious. The experiment was universally condemned as reckless, premature, and completely unethical. It just blew past all the known risks and violated international norms for scientific research. The CRISPR babies controversy really forced a global conversation that we probably needed to have. The cat's out of the bag. It's no longer a question of if we can edit the human germline, but should we? And that puts us at a massive crossroads, facing one of the most profound questions of our time. And that really brings us to the huge debate happening right now. On one side, you have a very strong call for a moratorium, basically a temporary ban on implanting any gene-edited embryos. The argument is, let's just pause until we know it's safe and until we as a society can figure out the ethics. But on the other side, some people argue there's a moral imperative to keep going, that to deliberately stop research that could save lives and get rid of horrible diseases is itself morally wrong. And so that's where we're left. For the first time ever in the history of life on this planet, one species, us, has gained the power to consciously direct its own evolution. We can literally rewrite the code of life. So the question is, what should we write? And maybe just as important, who gets to decide?